Okay, so human microbe, we probably will finish that up today, and then I'll get into a modified discussion on epidemiology. So you covered that a little bit in lab, and I have a little bit more I want to say about that in lecture. I am running behind according to the syllabus, so I'm going to be truncating some of these PowerPoints to fit everything in. Giving you the highlights. I think we left off right around the idea of here. I think it kind of makes sense that colorectal cancer, it's right in the gastrointestinal tract. It kind of makes sense after everything we've been learning about how microbes influence the human, that it makes sense that they could be involved in this as well, both in promoting it or helping it to never occur to begin with. And a lot of that is through the food choices that the human makes, and then the composition of the microbes, the ratio of the microbe, and what they're doing could either promote or not promote the colorectal idea. So I think we left off here looking at the standard American diet, processed, low fiber, typically high fat, high sugar, excessive animal protein, excessive fat intake, especially red meats, processed meats, could promote colorectal fiber to protect. Butyrate, I highlighted there. Remember, butyrate is one of the short-chain fatty acids that many microbes in your gut can make, and that's considered to be very beneficial for you and your cells of your intestinal tract. So they're saying that could prevent progression in colorectal get enough butyrate. Protein fermentation can ferment proteins. That is a thing. We do really talk much about that in lab. Tend to focus on sugar fermentation, like those lactose fermentation to glucose. But you can get protein fermentation that can too much of that going on, and it leads to inflammation. I think some experts have even classified red meat as a carcinogen. Have you heard that? Kind of scary thought. I think that's still up for debate, but some people have taken that stand. Some experts in the field of diet, nutrition, medical doctors. So just be careful about that. That's some thought. And again, abundance of fiber. Again, pointing to the idea of high fiber really is important. I was just having a discussion with a close friend last week, and she said she wanted to improve her diet, and she immediately started talking about protein. She wanted to increase her protein intake, and I said, well, why are you zooming in on protein? And it's mainly because so many diets, the ones that follow this diet, you'll lose weight, follow this diet, you'll feel great. It's a lot of high protein, and actually, at least some of the published studies I've read, we don't need as much protein as what a lot of those people are claiming. So I would encourage you to look more carefully at that. And then the other misnomer is that they say, well, if you go on a vegetarian diet, you don't get enough protein. It's like, well, but plants are organic beings. They're composed of protein and carbohydrates, lipids. They have protein. You're getting protein when you intake even plants. You don't have to do the red meat or meat in general. So it's a good idea to kind of look into this thing of diet a bit more carefully, because that's really the most intimate way that you have of interacting with your diet or with your environment is your diet. What you choose, because you're taking part of that environment and actually ingesting it, putting it in your body. And so that can have obviously a pretty profound impact. So the SAD diet, the Western diet. So anyway, going back to that conversation, I said, can I give a suggestion? She said, yeah, sure. I said I would focus in on fiber. Most of us are deficient in fiber, and that fiber, remember, fiber is like a prebiotic. It feeds the microbes in you in a good way. And so we'll see if she latches onto that idea. When COVID-19 hit, the first stages of the pandemic, I was shocked at how little research was being done on the role of nutrition in whether or not you get it and how severe of a case might you get if you ended up contracting. I was shocked at how few studies were being published in that time period, and even like 1 or 2 years into the pandemic. This was one of the first ones I could find. This was a 2021 paper, but 2021. Very few papers out there looking at nutrition. And to me, it just makes a whole lot of sense with all the microbiome research I've done, that of course what you eat could influence whether or not you'll get COVID-19 and how severe will that be for you. And so now they're starting to look a little bit more carefully. Of course, no one predicts, well, some people predict pandemics, but it's very tricky. So this research is coming after the fact. But this idea here that, here they're saying there's limited evidence that high dose supplements of micronutrients. So they're basically saying, really beefing up the proper micronutrients could be beneficial. This one was actually, I showed you this in the previous slide set, but I put it in with the idea. Again, the prebiotic idea. Inulin is a fiber. If you look at things, supplements that you can take that are labeled prebiotics, if you look on the ingredient list, inulin is common. It's a good one. And they're looking at that as a way to limit how severe the COVID could be, or whether or not you get it to begin. So they're starting to look at this idea because, some people never, the pandemic went and they didn't get COVID. Why did some people get it? Some people didn't. Some people got a very severe case. Some people died. Some people didn't. And I think the gut microbiome can go a long way toward this idea. The other thing too, it's not fun to think about, but when you think back to when the pandemic initially hit and we were told to sequester and kind of quarantine ourselves, that, of course, the microbiologist in me went, uh-oh, what is that going to do to our microbiome if we're all sequestered? And this is going to the idea of the hygiene hypothesis. I don't know if that's something that you've heard. That's been a term that's been thrown around here and there. They're starting to kind of phase that phrase out, actually, and they're starting to call it they're calling it, instead of the hygiene hypothesis, they're calling it the old friends hypothesis. The old friends hypothesis. Which, well, let me explain a little bit more before we get into that. So originally it was called the hygiene hypothesis, with the idea that if you're too clean, you're always cleaning your house, you're always using disinfectant on your hands, like the Germ-X type stuff, overwashing, and that you're starting to minimize your exposure to microbes, critical microbes that your body needs to interact with, your body may need to acquire. And if you take that to the extreme, when we were all sequestered for that stretch of time, we weren't interacting with others. We weren't interacting with a varied environment. And what is that doing to your microbiome? And here they're saying that, this is all occurring, this pandemic is occurring during the time when we're recognizing that there's a decade-long decline already going on in microbial diversity with excessive hygiene, too much antibiotic use, and then urban living as compared to being more rural, working on a farm where they're supposed to all the different animals, manure, and all of that. And so they said we're already dealing with a loss of a person. Then you throw this pandemic in, and we're all sequestered, and that's a double whammy. And physically separating from each other, too much cleanliness, not allowed to travel during the pandemic. And so they're saying all of this could really be hitting hard on what is it doing to our microbial diversity, and are we going to be able to recover from that? And I still talk to a lot of people who are still modifying their behavior, even in this year, 2024, with respect to the COVID idea, where they are still limiting how much they travel and who they interact with and all of that. So I think this is something that's not just during the first half year or year, but can still be in certain groups of people. It's still a thing. And so this is something that they're concerned about, that is going to really negatively impact our microbiome in the long run. It's a delicate thing because, of course, it's totally rational to be leery of COVID, certainly. And there are some key pathogens that are very deadly, including COVID, that virus. But there's, the vast majority of microbes around us aren't pathogenic, and they're actually doing beneficial things for us. So that balance of recognizing that, and that can be hard to recognize if you don't have any training in microbiology. You tend to only hear about the bad, the COVID and all of that. And you forget that, or you just don't know that there's all these good microbes here and in you and on you, and you need that exposure. In fact, the medical professionals are getting into this a little bit more rigorously and saying, they're even coining this term, microbiome first medicine, where they're looking at a way of treating the patient where you just don't look at the human. You look at the human with all its associated microbes and treating that entity. I don't know if you've heard the term holobiont. Holobiont. You heard that term? We're holobionts. Holobionts are you plus all your microbes that make up you, and that you are a superorganism. You are a holobiont, and that's a term that is being used more and more frequently now with regard to explaining the human condition. And I think this is awesome. I think that it's wonderful that the medical professionals really appreciate the impact microbes on us, and we need to look at that aspect of us as well as human physiology. Non-communicable disease and condition. So they're just saying, we've been throwing a lot of drugs at the human to treat this and that, and it's actually done some damage to the microbiome, especially when you think about antibiotics. So, to move towards sustainable healthcare, the human microbiome is front and center. So again, I think it's going to be the case where when you go to the doctor with some concern that you have about your health, you'll reach a point where they're going to be assessing your microbiome and looking at that. And they'll have enough information to look at your microbiome and assess if you're in a healthy state or if you're in a dysbiosis state, and then what to do about it. And hopefully there will be the right types of interventions, prebiotics, probiotics, other things that you can do. So I think it's really I feel very hopeful with all of this research coming along. And I hope that you will see it from that perspective too, that there is so much more that we can do now to improve our own health. Like, we have a lot more ability in our own. We don't have to go to a doctor and get a drug. We can do a lot on our own to improve our health. And hopefully the medical community will catch up on ways to help you. I think the microbiome has to be considered. So this is a great, in my opinion, this is a great way of moving forward, looking at how to help humans and improve their health. I bet you haven't heard about this yet. Microbiome First Medicine. It's not really out there yet, but I think you will. You'll hear about it in your lifetime. You'll hear more. So if you want to get to know your microbiome, you can. You can do it. You can go to a doctor if you want, but that's kind of pricey. I think I told you I did that with one of my sons, and it wasn't cheap. And of course, insurance won't cover most of that because that's too out there. Insurance companies don't get it. But anyways, this one's not too bad. This is on Amazon. I don't know. I haven't done any of these, but I was shocked that they're on Amazon's webpage already. Like, oh, I don't know how valid they are. Like, I don't know if they explain in more detail, like what it's actually testing, like how far does it go? You know, because you can go to family level of who's there, or all the way down to genus or species. And of course, the further specific you get, all the way down to species, the more expensive it gets. But I found these on Amazon's webpage. So most of these are fecal tests. You have to provide fecal samples and send them off, and which can tell you a lot, but not everything, because you know it's stuff that's coming out of you. It's not the stuff that's staying in you. And so they use it as a proxy of what's in you, and that's fair, but it doesn't tell you exactly like where they're located exactly. Are they in your small intestine primarily, or are they more in your large intestine? So there's some limitations, but it can be very valuable. Here's an idea: bugging our bowels, engineering bacteria to eavesdrop on gut health. This is actually a webinar that happened. I don't know if you put in that into a website search engine, if it'll come up that they've recorded this webinar. But they're looking at the idea of, and Tufts University is coming up with this idea of, could we get, like a pill that you could swallow and it could sample as it moves through. So it's like a little robot, essentially, that would take samples as it moves through your body. And then you essentially defecate it out and collect it and send it off for analysis. Doesn't that sound weird? So they are working hard on it. This is a real thing. Here's one of the images where you swallow the pill, and it's like this little nano robot type thing that actually will take samples of your intestinal tract as it works. And then it knows, each sample you can then analyze separately. You know exactly where the sample was taken from, whether it's small intestine, large intestine. So this kind of precision, they're working on it. I don't know. We'll see how far they get on it. But that could be another really interesting way to further get at spatial community dynamics. Where are they in your gastrointestinal tract? And that can go a long way to looking at, how they're interacting with each other and helping each other, competing with each other. So lots of interesting kind of cutting edge research going on. So it's still fair to say that you are mostly microbe. Like that t-shirt, I gave that to my nephew with graduation college. He's way into microbiology. Yeah, you're mostly microbe, even though it's not 90% like they originally thought. It's still over 50. Have you seen those stuffed animals? What's the name for them? I don't remember what the name is, but yeah, there's different microbes. I have this Diplococcus, and they're not showing it, but it has well. Former student gave that to me. It's Helicobacter pylori, because that causes ulcers. So, and one guy gave it. They're my friends. Okay. You ever want some summer reading that's really interesting and not too jargony? It's actually really an easy read. Ed Young does a really good job. He also does a lot of YouTube videos, I think, or TED Talks, where he's a science writer. This book, though, is all written about human microbiome. and it's very, very interesting. And then if you really are getting into microbiology, I went on to Etsy's webpage, into the vastness of Etsy. I was finding all kinds of funny things with microbiology. I don't know if you can read this one. And it's true, you're never alone. I have millions of invisible friends. I should say trillions, really. I have trillions of invisible friends. You're never alone. Even if all your friends ditched you on a Friday night and you're home alone, you're never truly alone. But yeah, there's all kinds of funny stuff. Be serious. You cannot be serious. Whoever, I can't remember who that person was who named that, but they intentionally did that. They had a sense of humor. Microbiology is my jam. My college career included alcohol streaking and antibiotics. Hopefully you'll say more about your college career than that, but these are actually, they look like teaching sites, freaked out. Those are soaps, hand soaps. But that would be teaching, virus plants, I like that one. And then socks and scarves and earrings. And then these 3 types of coasters, like drink glass. So all kinds of, I had no idea. Last time I looked for microbiology related stuff was years ago. So I was stunned at how much stuff was popping up when I did a search. The lanyard, so anyways, microbiology in your microbiome, hugely important for your health. Yeah, there's so much more to say. The published studies that are coming out, it's fast and furious and hard to keep up on. But I think you get the gist of what's going on with this type of research and some of the more exciting studies that they're following up and some of the findings that all that data is showing them. So fiber, lots of fiber, good for you. Any questions on the human microbiome? So, feed your microbes. All right. Let's see. Is it posted? Is the epidemiology slide set posted? Does anyone see it there? I don't see it. Do you see it on your end? No. All right, let me. Put that. All right, if you, oh wait, let me make sure. Okay, now if you refresh, you should see it in there. Is it showing up now? All right. Now, you've had a little bit of epidemiology concepts in lab. I'm not going to duplicate that. I'm going to focus on stuff you haven't had yet. And again, this topic is massive, so I'm giving you a taste of it, not certainly an in-depth review. I think this is one of the most challenging jobs in the realm of microbiology to have, looking at how to figure out what's causing the disease and then where did it originate, as well as making predictions on spread and how to control it and eliminate it. There's a lot to it. Now, if you flip it a little bit and think about this process from the pathogen perspective. So when a pathogen is interacting with you, their goal is to stay alive. They want access to your nutrients. They want a safe place to live. And they want to stay alive and reproduce so that they can get their genes into the next generation, and stay alive. So the pathogen's perspective is not really to kill you, because if you're dead, at some point the nutrients run out pretty quickly. And then if you're dead, you can't walk around and share the pathogen to other humans. And so from that perspective, it's usually not to the pathogen's best interest, so to speak, to be that deadly. And we'll talk a little bit more about that. So I'm thinking about the pathogens that need the host to interact with other humans in order for that pathogen to spread. Then you don't want to kill your host. You want that host to stay alive and be moving around enough that you can jump from human to human. And so that homework assignment on can we domesticate germs is getting at that idea. If we can understand from the pathogen's perspective what it's doing, and then if we take certain interventions based on their ecology and their evolution, you should be able to better manage certain very deadly diseases. So they go into some very nice, he gives some very nice examples on how we might be able to do that. Now, of course, from our perspective, we don't want to get sick. We want to keep the pathogen at bay or get rid of it totally, and we certainly don't want to die from it. So there's this perspective, depends on which perspective you're thinking about. If you think about the pathogens that get in you and persist, where it can be then labeled a chronic infection. Then there are the acute. So that's kind of the two major differences. You can have chronic or acute. Acute is usually shorter term, but usually more dramatic. Acute tends to be resulting from what we call emerging pathogens, which means they're new to us. We haven't seen them much at all. We haven't interacted with them in any time scale. And so when we get that first exposure, it can be quite dramatic, and often it can be deadly. So to give you some examples of different pathogens and some of their characteristics, I guess, you can see this table has some bolded sections. So this section here that's showing is what they call common source epidemics. And if you look at the source of the microflora, it's showing how you can get contaminated or infected with these different organisms. So, for example, botulism, you can get that actually from the soil. There's actually certain forms of there's different forms of botulism. Some are very deadly and others are fairly benign, especially if you go get treated quickly. And so the more mild versions, farmers actually can get the mild version and of skin, the cutaneous. And so that can be caught early enough. And then if you look at some of the other ones here, E. coli 0157H7, you may have heard that type of E. coli. That's usually manure or feces that is contaminated water or food. Talk a bit more about that later. Then if you look at the next part of the table, it's getting at host-to-host epidemics. So these are the ones where, you know, these pathogens don't really want to kill you because they need a host-to-host to spread and stay alive. So, you know, as most influenza cases, most people survive that. Not everyone, but who comes around and survive. And then sexually transmitted diseases, of course, again, that's not the simple idea. It's just specialized as through sexual interaction. And then you can see that there's vector-borne, where you have some kind of insect, but not always. So, usually insects, even if there's like a rat in the room. And then direct contact, what we know is contact with birds or birds. Like, so as you're looking at this, what I'd like you to do as you're studying at is know the bolded sections, like what are the potential, or could have common source. You could have host to host. You could have sexually transmitted. You could have vector-borne. So know the bolded terms. Those are different mechanisms. And then one example from each category in case I ask you, you know, like an essay question or a multiple choice where I ask you to choose. Okay? Or it might be more like a matching, I guess. Okay, let's say you are have the unfortunate situation where you do get exposed to a pathogen and it is successfully in you and making you sick. So that's the invasion step. Initially, you got to get invaded somehow, and then they have to set up camp and start to grow. And that's usually a time when you don't know you're sick yet in the initial stages, in the infection stage. And then you have the incubation period. This is where you are infected, but you're not really getting any overt symptoms yet. It really does depend on how much of the pathogen you got exposed to. So dose makes the poison is a really good thing to think about. The pathogen load is the fancy way to say it, where they look at, you know, did you ingest, like say you went to a buffet, you know, you went to lunch at a buffet and you were picking out different foods from the buffet table. And, you know, when you ate that piece of chicken, did it have 10 cells of salmonella on it or 1,000? And that can make a big, big difference to whether or not you're going to get sick. And it varies depending on the pathogen. There are some pathogens that literally 10 cells will make you so sick. Whereas with another pathogen, it can be well over 1,000 and you'll be like, I don't feel so good. But it's not that bad. And so it really dose makes the poison very much. Virulence factors, VF. What kinds of virulence factors are they pumping out into your body that you're reacting to? How quick is their life cycle? Are they super fast? Does it take them a while? All of that plays into how quickly you're gonna and how deeply you'll feel the symptom. Certainly your own health and your ability to resist the pathogen. And where does it get in versus where does it set up camp and make you sick? That can take a while. Like they might have to migrate a fair distance in your body. And so that could take a while. So all of that tends to go on during the incubation period where you don't really notice that you're sick yet. So this idea that different pathogens have different time periods of of the, you know, the initiation. Here you can see on the x-axis the days, and then they have the various. So influenza, you can see it's very short for past infection, hard to feel. But AIDS, on the other hand, it could be many years. So it really does vary. You know, and with each one, they have to figure this out. And the longer, you know, like rabies, it could be 14 days up to a year or two, really, before you start to kick into the next phase. So infection, it's getting in, it's starting to set up camp. Incubation, it's, you know, that's the next phase. And that can last from a few days to years. And then we get into the prodromal phase. That's the fancy word for you feel off, but you don't know what's going on yet. You're like, oh, I'm achy, or I just don't feel good. I'm extra tired, or my stomach's upset. So you get those mild symptoms. And then you kick over into the acute or acme period. That's where you kind of peak out. You're at the peak of the infection, and it's really the worst of it. And then if you survive through that, you go into a decline period, which means that you don't feel as sick, you're starting to recover, and then you enter into a convalescence. So they have this mapped out for all the different pathogens that infect us. And the one thing about convalescent period is that you need to be careful that you're not overdoing it during that stretch, because you have to appreciate, and it depends too on the pathogen, but the pathogen, I mean, they tend to do damage to your body. They're lysing your cells, they're pumping toxins at you. You have to recover from all of that. Your tissues have to replenish, you have to recover from all of that, and that takes a little while. So the convalescent period, even though you feel pretty good, you still have to be careful. So if you wanted to map it out, what we just went through in those words, this is the figure that they tend to show to map out those different phases of the pathogen interaction. A nice bell-shaped curve for the most part. So I think most of us could come up with ways that we can get infected. And, you know, we've lived through a pandemic, so we know that there's lots of different ways we could get exposed. Certainly host to host, but you can do all kinds of indirect ways. The fancy word that they often use for this idea of some kind of inanimate object that you happen to interact with. Maybe it's the bench surface or the elevator button or whatever. Fancy word for that is fomite, if it's an inanimate object. And then we tend to use the word vector if we're thinking about a living thing that is carrying the pathogen and you interact with it. So maybe it's a mosquito, some kind of insect, or a vertebrate that you interact with. And then sometimes they'll use this word, vehicles. It's a some carrier of a pathogen from its reservoir to us. So usually they're thinking about food or water. So think about like cattle that are dropping manure in a field, that then that manure gets into a nearby field where it's growing spinach for sale, and then the spinach gets sent on to eat it. That would be like a vehicle. So the reservoir would be the cattle back on the farm. This is more of a visual of what we were talking about here in terms of different ways that you can acquire pathogens. Have you heard, like people will say, I stepped on a rusty nail, and people go, you need to go have a tetanus shot? It's not the rustiness of the nail. That's not really the thing that's the problem. Really, the rustiness is just implying that nail's been there a while. But it's not the rust itself. Like I've had some people think it's the rust, causing some issues. I know just the fact that it's old nail and interacting with all the protein that you're in for a long time. What the danger is usually nails are fairly long and slender so that they pierce deep into your tissue, your skin tissue, and create some lovely anaerobic sites where anaerobes then can just flow. And so that's one that like is creating great opportunity that might be on the nail, like interact with that nail quite be there. But I think, if I were to ask you, most of you come up with a lot of this already, just living your life. And I think you've seen some kind of video or footage of what happens when you sneeze. That's something that they really tried to hit hard during the COVID pandemic. And to sneeze into your arm, if you can, rather than into your hand. And certainly just don't let it fly. This one is from your book. They took a kitchen sponge that you clean your plates with and your silverware, and they looked at it, and all the red over all my green and the sponge. Look at that. All those are all my. So if you use a sponge, you have to have ways of regularly cleaning and disinfecting it. I used to use sponges all the time, and then I switched over to more like a bristle brush. Those are easier to disinfect. They don't fall apart when you go to disinfect them. But yeah, he found a lot of stuff off that one sponge. Look at all the different microbes, the different colors in the pie chart. Lots of different microbes. Now, all of these pathogens? No. So again, it's not as big of a deal as, it's a little shocking when you see the data, but you have to remember a lot of those don't harm you. But it's something to keep in mind. So another thing that they look at when they're trying to assess what's going on with the pathogen and what is it, and they look at the kind of progression. So on the y-axis, we have the number of cases reported each day. On the y-axis, we have the number. And they'll match it up. And if you see this very quick uptick, like that very quick rattle like that, that's very classic for common source. Probably the most that way back in the 1800s when Dr. John Snow, he's like the father of epidemiology, and he found that a well pump in England was the water in that pump was there was cholera. I believe it was cholera. So that's a common source. People always went to that well for their drinking water, and people were getting sick, and he mapped it out. He could show this kind of onset. He took the pump off the well, and all the cases cleared up within a matter of days. So he was the first person really to say, no, there's a common source. That was great. On the other hand, if two fires close to intermingle with each other and have host-to-host contact, that takes longer. And so you see a more slow progression like that over a longer period of time. So these are like classic examples of being able to tease apart, is it a common source epidemic or is it host-to-host? Then you have to think about this idea of as the pathogen and host are interacting with each other, they do co-evolve with each other. We've seen that with COVID-19 and with the influenza and other, there's this co-evolution idea where you have, fancy way to phrase it is the process in which two or more species act as selective forces on one another, and each undergoes evolutionary change. That's the idea of co-evolution. And so with the COVID-19, that's why it's become less virulent. It has co-evolved and adapted to us. And then we've also co-evolved against it as to resist it better. So there's this idea of how we interact with each other and so undergo evolution. The classic idea of this is way back, they looked at rabbits as a host for the myxoma virus pathogen. So this virus goes from a mosquito bite. So the mosquito goes and bites the rabbit. The mosquito injects the myxoma virus into the rabbit. So this is the idea. This is going to, the classic study is rabbits were overrunning Australia. And they didn't know what to do. And you know how quickly rabbits can reproduce. So there's like, having hunting season on rabbit wasn't doing it. They couldn't keep up with it. So they said, let's introduce the myxoma virus into the rabbit population. And so they deliberately did this. And initially, the virus was getting spread to the rabbit, and the rabbits died. And so they're like, okay, this is working. We're getting a downtick in the rabbit population, just like what we want. So this is working. However, after the first year, so the first year was successful. 95% of the infected rabbits died. They're like, okay, this is a good way to control the rabbit population. However, by the end of that first year, when they re-isolated the virus at the end, they showed that it wasn't as virulent as what they started when they started this experiment over in Australia. In addition, the rabbits were showing increased resistance to that virus. And so they both, the myxoma virus and the rabbit, had co-evolved with each other. And by the end of the 6th year, we only had 4%, which is still, a lot. But what happened is they kept following that study out year after year after year. The rabbits gained resistance and they repopulated. So it was pretty much a failed experiment. Initially, the idea, made sense unless you thought about the coevolution process. Then things got really muddy really quickly. So this is the data plotted out where they're showing that as the virulence of the virus is decreasing, the mortality decreasing. And so you have this idea of coevolution going on between the pathogen and the host. So that did not work in terms of their wanting to decrease the rabbit population. Oh, all right, we'll stop there. I have a few more things to say about epidemiology, but we'll do that for Wednesday.